Today, we are going to explore the concept of electrophoresis. So what exactly does electrophoresis mean? Electrophoresis, or electro, meaning electric field, and phoresis, meaning migration, is the method of using an electric field to separate molecules based on their size and polarity. Here, we will be utilizing gel electrophoresis as a means to separate samples of DNA molecules. DNA is a negatively charged molecule that can be cleaved into different sized fragments using molecular scissors called restriction enzymes. In this demonstration, we'll be separating 4 DNA samples, each containing millions of individual fragments. These fragments can be one or more different sizes of nucleotide base pair lengths. Sample A contains DNA fragments of the same size. Each fragment is 2,000 base pairs. Sample B also contains DNA fragments of the same size. Each fragment is 1,000 base pairs. Sample C contains DNA fragments of 2 different sizes. One has 300 base pairs and the other 500 base pairs. Sample D contains DNA fragments of all 4 sizes. With the samples ready, it's time to start setting up the gel electrophoresis chamber. In this demonstration, we'll be working with the Mini 1 electrophoresis chamber. We will begin by plugging in the unit. With the lid removed, a precast agarose gel is placed into the tank. Agarose is used as a medium to separate molecules because of its porous, spongy matrix. Next, 135 milliliters of running buffer is added to the tank by pouring half of the buffer into one side of the chamber until the air trapped under the tray is pushed out. The remaining buffer should then be poured into the other side of the chamber until the gel is fully submerged. The buffer is full of dissolved solutes. Its conductivity creates a uniform distribution of ions from one electrode to the other through the gel electrophoresis chamber. As the electric charge flows through the agarose gel, it will mobilize the DNA samples, causing them to move. Since opposites attract, and the DNA is negatively charged, it will begin to migrate toward the positive electrode. At this point, the low-intensity blue light can be switched on. This will make it easier to see the wells in the agarose gel to load our DNA samples. Wells are cup-like depressions that are created by use of a comb when the gel is cast, and are used as the location for which to place the samples for separation. The number of wells a gel has can vary based on the comb selected. As you can see, our gel has 6 wells. Because we have just 4 samples, we'll only be using 4 of those wells. Using a micropipette, the DNA samples are carefully added to the wells in the agarose gel. During this step, be very careful when inserting the tip of the pipette. It should only be lowered about halfway into the well to avoid penetrating the bottom. Be sure to record the order in which the DNA samples are loaded. It is also very important to change the pipette tips between each sample to avoid cross-contamination. With the gel loaded and the lid replaced, the unit is ready to be switched on. As the current is applied, it flows from the negative electrode to the positive electrode. Since DNA is negatively charged, it migrates toward the positive electrode through the agarose. Loading and tracking dies have been added to the samples. This die makes it easier to load the samples since DNA is virtually colorless, while also creating a die front ahead of the migrating DNA. On the molecular level, the matrix of an agarose gel looks a lot like a sponge full of pores. As the DNA molecules migrate through the gel, they will encounter obstacles and bump into structures. We can imagine the agarose matrix acting like a series of barriers that the DNA has to pass through. The length of the DNA fragments has a direct correlation to the speed at which they are able to navigate the agarose. This means that the smaller the molecule, the less it will collide with the agarose, allowing it to move through the gel more quickly. By comparison, the larger the molecule, the more often it will collide with the agarose, causing it to move more slowly through the gel. As a result, The smaller molecules slip through the agarose faster than the larger molecules. As the molecule bands move through the gel, they are led by the tracking dye. This dye front migrates the fastest through the agarose and serves as a visual indicator for when to stop the electrophoresis process. Once the dye front reaches the end of the gel, the power is cut off. The agarose gel is now ready to be examined and the DNA banding patterns analyzed. Each of the individual bands represent millions of pieces of DNA of similar size that are stuck within that section of the agarose. Compare the loaded samples to the banding pattern for analysis. The analysis of DNA banding patterns, like those demonstrated in this video, can be used for a variety of applications, including forensic science, food safety, paternity testing, and much more. Special thanks to Mini One Systems for their support in making this video possible.